Really be interesting, because I don't think we know that much about embryonic mitochondria. So, but I was curious, how do you think that they proliferate? You said they proliferate under stress. But what's the signal? Because PGC1 is not important in the embryo. So do you know what the pathway is there that drives their proliferation?
First of all, I'm not a specialist in mitochondrial biology. I'm a gynecologist, so I can speculate, but I really don't know the answer to that.
Other questions? Could I, in a minute, so I'll see your hand, but could I ask you, so you showed us some of the augmented data that's presented but not published as far as I... Okay.
Not published, no clinical registering, clinical trials.
Because it got a lot of attention in the lay press. Are you aware of other similar type of approaches, sort of to rejuvenate the embryos?
Yes, I do, but not with mitochondria. I mean, this is another issue altogether. As I told you, you know about the work of Kawamura and Aaron Shue trying to increase the number of follicles or to, as I said, to awake these dormant follicles. We have already a pregnancy also using a similar approach because they have been treating the ovaries with cancer, pro-cancer substances, but just because of our experience in fertility preservation, we did a different approach, and we have already one pregnancy. We have also a pregnancy stimulating these dormant follicles with stem cells from bone marrow, injecting bone marrow cells near the ovaries. So there are different approaches. And also, we are also working in the lab trying to repair these DNA breaks. So in this whole chart or picture of aging, there are many ways you can approach the age-related changes. I just discussed the mitochondria because the topic of the meeting was stress, and also in the interest of time.
That's clear. Last question, yes, please, Rob.
Yeah. So thanks, Tony. That was really an interesting review. So I guess another interpretation would be that the mitochondrial biogenesis is being driven by injury, essentially. So the reason that you're seeing more in the aneuploid or sick embryos is that biogenesis is part of that. And mitochondrial DNA doesn't have it's not protected by histones, and the mitochondria is where actually all the oxygen-free radicals for the whole cell are being generated. So the oxidative stress inside the mitochondria has to be higher than it is in any other compartment. And you've got all those guanosines, which are very sensitive to oxidation. It would seem to me that that's a place where you're going to get a lot of oxidized DNA damage. You mentioned a little bit about DNA repair mechanisms. Maybe those are super efficient or something, but I would imagine that maybe just having more mitochondria might not mean that they're healthy or doing the right thing.
Thank you.
Okay, I think we've got to stop here. Thank you once more.